So we're going to talk about dimensionality reduction and visualization of single-cell RNA-seq data sets. So again, looking through our clustering workflow, yesterday we talked about normalizing expression values. And then after this lecture, we're going to do the lab for normalizing expression. But because of how the timing works out, it fits much better if we do that today with our visualization lab, because the visualization lab is very short. So this lecture, we're going to talk about feature selection and dimensionality reduction, as well as visualization, which many people confuse with dimensionality reduction, but hopefully by the end of this lecture, you'll know what the difference is, and you'll understand what I mean by data visualization versus dimensionality reduction. So to start off, we have to first understand what the problem is we're trying to solve. So the problem in terms of our data is what's known as the curse of dimensionality. And the idea here is that for our single-cell RNA-seq data, each gene counts as one dimension. So if you see this one-dimensional line over here, up here, that would be the expression of one gene. So that might be the expression of GAP-DH. We can then go to the two-dimensional point, and then we might have the expression of GAP-DH and the expression of ACTA-B. Then we can go to three-dimensional case, and here we might have the expression of GAP-DH on one dimension, ACTA-B on another dimension, and P53 on another dimension. And you see, as we're going up dimensions, the total space that our points are living in is getting bigger and bigger. But the number of data points in our data set is the same. So we've got our finite set of cells that we're always looking at, and as we're increasing the dimensions, so the number of genes we're looking at, we're getting it more and more space for those dots to be in, so they're going to get more and more spread out. Another way to think about this is that each dimension, so each gene, has some noise, right? We measured the expression of these genes using sequencing technology, and that technology has some random noise to it, right? We're just randomly sampling molecules from our sequencing library to sequence. We're randomly hybridizing our RNA molecules in our droplets. There's some inherent noise to that technology when we're measuring every gene. So every gene has some noise in it. However, the effect size, so how different a T cell is from an epithelial cell, stays the same, regardless of how many genes we're looking at. This means as we increase the number of genes we're looking at, the noise increases while our signal is staying the same, and eventually that noise is going to swamp out the real signal. And it'll be really hard to tell what our two different cells are from each other. Okay, so in single-cell RNA-seq, we're measuring thousands of genes, or tens of thousands of genes in many cases. All of those contain technical noise from our sequencing experiment. They all contain biological noise, right? Cells aren't perfect. They're sort of randomly transcribing genes as polymerases randomly bind to the DNA. But only some of those genes are responding to biology, right? We've got lots of housekeeping genes. They're just going to be sitting there, expressed everywhere at sort of a uniform level. We're not getting any information about what cell type is what, or what the cells are doing based on those genes. So what we want to do when we're doing our downstream analysis of trying to find cell types or trying to find trajectories in our data is we want to look at just those genes that are actually responding to the biology, right? Or they're actually doing something relevant to our question. The question is then, how do we find those genes out of the 10 or 20,000 that we measured? So what does an interesting gene look like? So here's some single-cell RNA-seq data from some B cells. So you can see 3 genes that are part of the B cell receptor. So we've got IgHG4, IgHG1, and IgKC. Those are all components of the B cell receptor. And they're very differentially expressed between different B cell clones. And then we have a housekeeping gene RPS3 here, that's just sort of universally expressed everywhere. It's just a ribosomal protein. So clearly, just by looking at these plots where each dot is a cell and they're colored by the expression of those genes, you can see the interesting genes that are actually defining our cell types look quite different from our housekeeping gene. So one of the ways they're different is they have a high variance across the cells compared to the housekeeping gene. They're also often correlated with each other. So IgHD4 and IgHD1 are highly correlated with each other in this case. And they're related to the known biology of these cells, right? They're the B cell receptor, these are B cells. So they're clearly important to these cell types. So if we want to use these characteristics to try and find our interesting genes, how could we go about doing that? So if we just look at the high variance genes, we have a problem in that in our single cell RNA-seq data, the mean expression of a gene and the variance of that gene are related to each other. So here's the plot of a bunch of genes in these cells, where we have the mean expression on the x-axis, the y-expression, the y-axis is the variance, and you can see some of the cell type defining genes here colored in red. So these are the genes we want to pull out. If we just use a threshold on the variance, right? If we drew our threshold down here to get all of those red genes, we're also getting some black genes that aren't relevant. If we go more strict to get just those genes, just red genes and none of the black genes, well, now we're going to miss some of the red genes, right? Because we've got this increasing trend. So some of the highly expressed, not relevant genes will have a variance higher than the lowly expressed relevant genes. We could look at the gene-gene correlations across all of our cells and all of our genes, but that takes a really long time to calculate. So for that, we have to compare every gene to every other gene. So that's immediately 20,000 things times 20,000 other things. And we also have to look at their expression across all of the cells, which could be another 50,000. Right? So that's just going to take a lot of compute power to do. But we could do it. We could look at those related to the known biology, but in many cases, we don't actually know what the biology is ahead of time. Otherwise, why are we bothering to do the experiment? We already know everything that's going on. We don't need to do any more experiments to find out. So we could also think about what biological features really are. So in many cases, when we think about biological features of our cells, we're not thinking about individual genes. We're thinking about things like what metabolic pathway is active? Is the notch signaling pathway active in this cell? Is the cell involved in inflammation? Does it have a cilia? Is it senescent? Right? These are all combinations of multiple genes. But we have a problem in that we don't know what all the relevant features are, and we don't know which genes contribute to these features. Yeah. So our two solutions to the curse of dimensionality, one is known as feature selection, which is where we're going to choose which features we're going to use for our data set. And for single cell RNA-seq, what we're actually going to do is we're going to use the variance. But we have to first fit a relationship between that variance and the mean expression to correct for that relationship. But that's fairly easy to do. So that's what we're going to be doing when we do highly variable gene selection. The other way we can reduce the dimensionality is what's known as dimensionality reduction, or feature extraction, you'll also hear it called. And this is where we create a new dimension from the combination of existing features. So this might be, we're going to take all of the genes related to the cell cycle and compress them down into one feature that's just representing the cell cycle. So there's two ways to go about this. The first one is what's known as supervised, and this is where we're using existing biological knowledge. So we can go into the databases like the gene ontology find all the genes related to the cell cycle, and then use those to build our new feature. By saying, okay, all of those genes in the cell cycle, we're going to now average your expression, your new feature called cell cycle. So the main method for doing that is called GSVA, and it works reasonably well. There's also an unsupervised method where we're going to say we don't know what genes do what, but we'll use the correlations between genes to figure out which genes are involved in the similar process. So there we're going to look at the correlation between all of our different genes and see, is there a group of them that are all highly correlated with each other? Okay, we'll merge those together and make a new feature out of those guys, because they're probably involved in the same process. So the main method people use for that is PCA. So the main problem with the supervised method is that our annotation databases aren't that great, and many of the processes are incomplete or missing, and we don't know all of the genes involved in all of them or all of the processes that exist in our cells. So for the most part, we're going to be using unsupervised PCA for our dimensionality reduction.
We have a question in the Slack. If we are compressing all of these genes down into one feature, doesn't this mask so much relevant biological information?
That's a good question. So yes, it is somewhat masking biological information, but it's also removing noise. So the idea with especially with principal component analysis, is to keep as much of the relevant biological information as possible while also reducing the noise in our data. But yeah, we will be losing information when we do this. So it's principal component analysis. So for principal component analysis, or PCA, we take our cloud of data and we try and draw straight lines through that data where each of those lines is capturing as much variability in our data as possible. So in this case, we have our data set over here, and the first principal component that captures the most variability is going this way through the data. And then we find our next principal component that has to be at right angles to this first one. So we essentially take an arrow and go around in a circle orthogonal to this first line and find the direction where it's capturing the next most amount of variability. So in this case, it's sort of going this direction. And you can keep doing that as many times as you like, up to the maximum number of dimensions in your original data set. However, because we're capturing the most variability in our principal components in order, it means if we keep the top few principal components, we're going to be keeping as much real biological signal as we can. And the lower down principal components will be capturing mostly random noise. So we can throw those away to get rid of the noise and keep the ones at the top that should be capturing our biological variation. The problem with principal component analysis, though, is that it assumes our data is normally distributed.
We do have a follow-up question in the Slack. Let's say I'm interested in cell cycle signaling. If the goal is noise reduction, wouldn't it make sense to remove or reduce dimensions not related to cell cycle genes, but leave those related ones, quote, unreduced, so we can still examine their individual response?
Yes. Yep, that absolutely makes sense. And one of the things that I actually recommend doing is when you do, especially things like highly variable gene selection, if there's certain genes or processes that you're particularly interested in, check whether they're in your highly variable gene list. And if they're not there, add them to make sure you're keeping all of those genes that you're interested in.
Okay, thanks. And we have one more question. What should we do if the top two PC axes do not account for enough variation in the data? For example, the variation is uniformly distributed across many PC axes.
That's another good question. There you're going to be keeping more than 2 principal components. So in single-cell RNA-seq, usually you need to keep at least as many principal components as there are cell types in your data set. Sometimes you need to keep even more than that, because there will be important variability in all of those axes. And it's also why most of the time we're not looking at our data in PC space, because the first 2 principal components are not going to be showing all of the interesting features of our data. And we'd have to make plots of the top 20 principal components to really see all of the interesting aspects of our data. And now we've got a giant page full of PCA plots.
Okay, and we have a couple of follow-ups here. And of course, I have lost my Slack.
Yes, the next one is, does that mean you cannot give a proper visualization downstream then? Sort of. We're going to get to that. So how we can do visualization and how proper that visualization is going to be. So the main problem with PCA is that it's assuming our data is normally distributed, and our single-cell RNA-seq data is not normally distributed. It follows a negative binomial distribution. So this means we have to do our log transformation or our Pearson residual transformation before we do our PCA, because both of those will make our data much more normally distributed, which will allow PCA to actually work properly. There is one other option, which I'm going to mention, but I'm not actually going to recommend, which is Z-wave, which is specifically a modification of PCA so that it uses a negative binomial distribution and is applicable to single-cell data. The main problem with it is that this method was invented in the early days of single-cell RNA-seq, where we had data sets of around 5,000 cells, and it doesn't scale all that well up to data sets of 50,000 cells. So if you have a small data set, you can absolutely try it Z-wave, and it might work much better for you than regular PCA. But if you're using a giant data set, it might take 24 hours or more for Z-wave B to calculate its principal components. But for SmartSeq2 datasets, actually, because we had a question about SmartSeq2, Z-wave B works really well for SmartSeq2 datasets. So you might want to consider that. Okay, so that's dimensionality reduction. Now we're going to talk about visualizations. So the goal of visualizations is not necessarily to preserve anything about our data. Our goal with visualization is to make a pretty picture that will give us some sort of insight about what's going on in our data. So whereas one of the nice aspects of PCA is that the distances between points are preserved in PCA. So if 2 points are close together in a PCA plot, they're actually close together in the real gene expression space. We're going to see with our visualization methods that that's no longer necessarily true, because the goal of our visualization method is to show as much of the structure of our data set at once in 2 dimensions so that we can make a plot to put in our paper. It's not to preserve the fidelity of our data to the real gene expression space. Right. So to sort of understand what's going on here, we're going to go back to 3 dimensions, because 3 dimensions our brain can handle. 20,000 dimensions, our brain can't imagine what that looks like anymore. So if we think about 3 dimensions, and we have a 3-dimensional cube that we want to make a 2-dimensional plot of, right? We want to make a nice plot of our 3-dimensional box for our paper. How are we going to do that? So we could do PCA. So if we do PCA, we're just going to draw some straight lines through our box and then project our box onto those lines. So here we'll have our 3 principal components like this, and we'll get PCA plots that look like this. Right, so all of our first two, we can see kind of the two sides, and then 1 versus 3, maybe we see the top of the box. So all of the distances between our sides of our box are preserved here, and the shape of our box is preserved. And if we think really hard about our principal component projections, we could logically infer from those images that we have a three-dimensional box. We can't actually see all the sides of our box at once, right? We have to have we have to have two different plots just to get the idea that this is a box. What we could do instead is we could rip open our box and unfold it onto our flat space. Right, so now we can see all of the sides of our box, so we can see all of our data at once, and it's all in one plot. But the side effect of this is that the distances between stuff in our plot aren't the same anymore, right? So this top edge here looks like it's very, very far away from this bottom edge. But in reality, in our 3D case, those would actually be touching each other, right? So we've lost some information, some fidelity of how our points are related to each other, but we can at least see all of our data at once. So this is more or less what's happening when we do something like UMAP or t-SNE. As it's going to unfold our data so that we can see all of our different cell types at once, but it means the distances between those cell types will get distorted. Different visualizations choose to show different aspects of our data. So they choose to preserve some aspects more than others. So here I've got the exact same data plotted with 4 different methods. So one is PCA, which we've covered. The other is t-SNE. t-SNE stands for T-distributed stochastic neighbor embedding. It's a long word that it's just related to the math behind it, but I'm not really going to go into that because it doesn't, in the end, it doesn't really matter what the math is. But t-SNE tries to preserve the cluster structure of your data, and its other assumption is that the density of dots in your plot should be as uniform as possible. So you can see already in this plot that the points in the t-SNE are more spread apart from each other than in some of the other plots. Whereas UMAP, which stands for Uniform Manifold Approximation Projection, or something like that, again, that's just related to the math behind it. It doesn't really matter for using it. But UMAP, it's based on links between cells, especially local links between cells. So it tends to create very dense groups where you have cells that are close together, as well as sort of strings between groups of cells. That's its main assumptions. The other one I'm showing here is a diffusion map. So diffusion maps specifically try to capture trajectories in your data. So if you have like a developmental trajectory of one cell differentiating into some other cells, diffusion maps tends to capture that better than the other methods. Because that's the whole idea that it's trying to preserve, is it's trying to preserve trajectory information. Right. Yeah, so PCA preserves accurate distances between points. UMAP aims to preserve the overall structure, so large scale structure of your data. t-SNE tries to preserve distinct clusters, and diffusion map tries to preserve trajectories and gradients. So now that you've seen these 4 different projections of this high dimensional data, what do you think the actual data looks like? The real high dimensional data in this case. I'll give you a few minutes to think about that. Ottawa, you can put your guesses in Slack, or you can just think about them and sketch it out for yourself.
Not sure if you've got the Slack in front of you, but we do have one answer from Ottawa. You can't really say since the dimensionality is too high, right?
Yeah, so it's hard, right? Each of these is trying to show you what your data really looks like, but it's really hard to know what it really looked like. Fortunately, I created this data set, so I know what it really looks like. So this is what it really looks like. The red and blue groups should be parallel to each other, and the purple group should be coming out at right angles to the red group in a completely different direction from the blue group. So that's what this data really looked like in high dimensional space. So now you can look at these different visualizations and see which one was the closest to correct.
Diffusion.
We've got votes for diffusion maps on Edmonton's side. Ottawa, we've got one vote for the T-SNE being most accurate. And I'm voting for UMAP.
Depends on your goal, I guess. Yeah.
Yeah, it's hard, right? They're all kind of capturing it, but also they're all kind of wrong. Yeah, few more votes for UMAP. Oh, we've got a question from UMAP. How did you determine what the data actually looks like? I created this data. So I created these patterns in 2-dimensional space and then projected them into 20-dimensional space and then calculated the PCA and diffusion map and T-SNE on them. So I know exactly what they originally look like. Yeah, so the point is here that it's hard to know which one's best. And overall, there's been lots of papers trying to debate which is the best one. And the answer is always, well, it depends. For some data sets, one of these visualizations will be more accurate than the other. For another data set, it might be a different one. So if you're trying to understand your data, you should try lots of different visualizations. There's no reason to just do a UMAP or just do a T-SNE. You can do both. You can do a diffusion map as well. You could look at the top 5 principal components and then try and piece together from all of those what the truth is about your data set. And then when you make your paper, you pick whichever one looked closest to what you think the truth is. Right, so that's how come you have different projections being used by different data sets in different papers. So here's another example. So here's a UMAP of a data set on the left. I want you to look at those 2 clusters in sort of cyan and, I don't know, mustard yellow, I guess, with the black arrows at them. Would you conclude that those 2 data sets are those 2 clusters of cells are similar to each other based on this plot?
Just look at blue and mustard.
Yeah, the 2 with the black arrows pointing at them. So do you think those 2 clusters are similar to each other based on this plot?
No.
Got some votes for no. Ottawa's just going, you don't know. So what if I showed you this one? And are the 2 clusters in this data set with the black arrows pointing at them similar to each other? No, so here they look very, very different from each other. This is exactly the same data set. Plotted just with UMAP on the left and T-SNE on the right. So you can see in the UMAP, those 2 clusters look much closer together than they do in the T-SNE. We could take another example of this blue and green group, where on the left in the UMAP, it looks like they're very, very similar to each other, whereas in the T-SNE on the right, they look more distinct. Right? Neither of those is objectively the right answer. They're both just different ways of looking at your data. So they're both correct. They're both equally correct and they're both equally wrong. Yeah?
I have a quick question. Because, for this kind of the clustering, they are clustered based on the gene expression, right? So we already know that they are different somehow about the variation of the genes and the machine is just trying to cluster them differently. But just a matter of using different, dimensional reduction is that how these two are close or far from each other, right?
Yeah,
But already that we can trust that they already clustered in two different populations.
we'll come to that when we do clustering. So the question Edmonton was asking was, because we did clustering, don't we know already for sure, because the clustering is done on the gene expression, or actually it's done on the PCA space, don't we know already that these clusters really are different from each other? And I said, we'll get to that when we do clustering, but that's not necessarily true. But yes, it is more evidence that they are truly distinct from each other, that they're clustering separately. But there are cases where you can get things clustering separately that are not actually different. So here I'm just as another example of ways visualizations can tell you different stories depending on how you use them. So these are both UMaps, both the left and the right. They're both UMaps of the same data, but UMap has a parameter. In fact, all of these methods have parameters to them. So tsne also has parameters. And here I changed the parameter, which in this case is k for KNN, which again, I'll talk about more when we get to clustering. But here on the left, I'm using a low value for the parameter for the UMap. And on the right, I'm using a high value. Right? So that's the only difference between the plot on the left and the right. And you can see, if you look at the clusters with the black arrows, that what you might conclude about those clusters would be different depending on what value of the parameter used. Right, on the left, you might conclude that these are two very distinct cell types, but on the right, you might say, oh, these are actually three cell types in a trajectory. So again, it's worth exploring lots of ways of visualizing your data when you're still trying to understand it. And now that we've talked about how UMap works and looked at some of how these things work, this is a UMap of a three-dimensional thing. What do you think it was?
Frog.
So Edmonton's voting for a frog. Any other guesses? Ottawa's voting a gecko. Someone else is voting a woolly mammoth.
These are like
Yeah, we got another vote. I got a vote for a lizard.
salamander.
Hmm?
Salamander.
Salamander. Got a vote for salamander. Oh, we got a vote for a turtle from Ottawa.
Could it be just a sphere?
Oh, possible it's just a sphere. In Edmonton, we'll see in a minute.
Symbol.
Disassembled sphere. Another vote for a frog from Ottawa. So it was an elephant or a woolly mammoth. Yeah, so it's a skeleton of a woolly mammoth from a natural history museum. Yeah, Alex from Ottawa got the right answer.
That's impressive. I didn't see it.
Yeah, so you can see here in yellow are the tusks. Here we've got the head. Here's one arm. There's the other arm down here. One leg over here, and the other leg kind of goes over here. And then we've got all the ribs and spine in the middle, sort of splattered.
Has anyone ever gotten that correct before?
No, this is the first time someone got it right. Very impressive. We've, frog is usually the most popular answer. So everyone voted frog, can rest in good company. Lots of other people think that as well. So we've got a question from Ottawa for what the K parameter is for UMAP plots. So UMAP is based on linking cells together in a graph, and the K parameter, Alex cheated because he has the PDF that shows it.
Oh, no.
The K parameter for the UMAP. The K parameter for the UMAP plots controls how many cells each cell is linked to. So it's sort of controlling the scale of your UMAP. So a high K parameter links cells to many other cells, which includes cells that are farther away from it. Whereas a low K parameter links cells to a fewer number of cells, and mostly just the cells that are very, very close to it. So that's what the K parameter does for UMAP. Yeah, but you can see very clearly that we're getting some information from our UMAP, but we're not getting the true full story. So you have to be quite careful interpreting it. And most of the time, you should do some sort of statistical test in PCA space or gene expression space to really draw your conclusions from. I wouldn't recommend just looking at your UMAP and saying, yes, this is definitely what's happening. Okay, so to summarize, single-cell RNA-seq is high-dimensional noisy data. We can do feature selection using highly variable genes to identify those genes with biological differences, not just noise. We can do dimensionality reduction to condense multiple genes together into a single feature to extract the main biological signals within our data using PCA. We can use t-SNE and UMAP to visualize the data. UMAP is super popular right now because t-SNE, when t-SNE first came out, had a bunch of problems with it that made it not as good as UMAP. The modern version of t-SNE is just as good as UMAP overall. So you can use both of them interchangeably for whichever tells the story you believed is true about your data the best. And these should be considered visualizations, not dimensionality reduction, because they're not preserving things like the distances between your points. Right? They are distorting your data. And the parameters you choose for UMAP and t-SNE can drastically affect the interpretation. So I have any questions? So Ottawa asks about t-SNE's reproducibility issue. So t-SNE, both UMAP and t-SNE involve random number generation. So both of them are not reproducible unless you set the seed. And the new t-SNE is much more reproducible than the old t-SNE. So there was a lot of reproducibility issues with t-SNE originally, but they all stemmed from the fact that the first version of t-SNE, so the way t-SNE and UMAP both work is they first make a rough guess of where your dots should be in two-dimensional space, and then they iteratively improve on that guess. The original version of t-SNE, its first guess was just randomly throw the dots onto 2 dimensions. Whereas the first version of UMAP was, let's do PCA first and use the first 2 principal components as our initial guess. So that using the first 2 principal components of PCA first made it a lot more stable because every time it was starting at the same place. And it made it much better because its first guess is actually pretty good, right? It's capturing the major signal in the data. Whereas t-SNE's, let's just randomly throw it wherever we want on 2 dimensions, was not very reproducible. Each time you ran it, the initial state would be very different. And it doesn't really reflect the data at all. So it would have a harder time getting a good visualization than the UMAP did. since about 2019 or so, people realized that this was the problem, and tsne from packages that were updated after 2019 now uses the same starting point as UMAP. So it uses that first two principal components, and now it's just as reproducible as UMAP, and it's just as good at making a visualization as UMAP. So that's why it was believed tsne has reproducibility issues, but it's no longer the case. Ottawa also asked, does diffusion map reduce dimensions or not? Yes, diffusion map also reduces dimensions. You specify how many dimensions you want. Usually people go for 2 or 3. It's again, it's sort of in between a dimensionality reduction method and a visualization method, because it's trying to preserve some degree of distances in the data, but it's very focused on trajectories. So it's quite good at representing trajectories. It does not perform well visualizing clusters. So it's really only used when you're looking at trajectories in your data. But it also reduces the dimensions. So when you run diffusion maps, you specify how many dimensions you want, and it will generate the best however many dimensions you ask for.